Hi, good morning. In this video, we are going to learn about the special details of bacteriophages. So, structure of the bacteriophages. What is bacteriophages? Phase means virus. Phase means virus. Virus infect the bacteria is called the bacteriophages. Virus infect the bacteria is called the bacteriophages. Generally, these bacteriophages infect the E. coli bacterial cells. That's why these are called the coliphages. Coliphages. Phase means virus. Virus infect the bacteria is called the bacteriophages. These bacteriophages generally infect the E. coli bacterial cells. So that's why it is called the coliphages. These coliphages are divided into two types. These coliphages are two types. T even phases and T odd phases. And even numbers and odd numbers. T even phases example is the T2, T4 and T6 etc. T odd phases example is T1, T3, T5, T7 etc. is the examples of T odd phases. Coliphages is classified into two types, T even phases and T odd phases. T even phases examples, that means even numbers, example T2, T4, T6. T odd phases example T1, T3, T5. These are the examples of T odd phases. In this video we are learn about special details of the T4 Bacteriophages. So in this video we are learn about details of the T4 Bacteriophages. So it is the T4 Bacteriophages. What is the T4 Bacteriophages? These T4 Bacteriophages shape is the tadpole shaped. T4 Bacteriophage shape is the tadpole shape. So in this T4 bacteriophages mainly consist of three parts. That is the hexagonal head part and middle collar part and tail part. Hexagonal head part, collar part and tail part. These are the three parts of the T4 bacteriophages. So in this head part, it is the head part appear hexagonal head part. It consists of hexagonal head part. The size of the head part is the 95 into 65 nm. Nm means nanometer. It consists of hexagonal head. The size of head is the 95 into 65 nm. That means nm means nanometers. In this head, it contains 50 micrometers long. 50 micrometers long double stranded DNA. 50 micrometers long double stranded DNA tightly packed inside the head. 50 micrometer long double stranded DNA tightly packed inside the head. It appears like a yarn of ball. So in this T4 bacteriophage, the genome is the double stranded DNA important. T4 bacteriophage genome is the double stranded DNA. Inside the head it contains tightly packed double stranded DNA. And surrounded genome consists of capsid. Surrounded genome consists of capsid. This capsid is made up of 2000 capsomeres. Capsid is made up of 2000 capsomeres. Capsomeres means capsid subunits is called the capsomeres, and this capsid is the present surround the genome. This capsid is again made up of subunits, that is called the capsomeres. In this T4 bacteriophage, the capsomeres number is the 2000 capsomeres. And next the middle part is the collar, These collar connect are joined by the head with one side and tail with one side. So collar is connected by both head and tail. So it connected one end of the head and one end of the tail. This collar also covered by gelatinous sheath. This collar is covered by one of the proteinous sheath. This sheath protect the protect the head part, and this sheath is covered the around the collar, and this sheath consists of protein subunits, that is the 144 protein subunits. Here consists of 144 protein subunits arranged in 24 rings, 144 protein subunits and arranged in 24 rings, and each ring consists of 6 subunits. So totally here arranged in protein subunits 24 rings. Each ring again consists of 6 subunits. Totally subunits is the 144 subunits. Okay, and next the third part is the, here observe, base plate. The third part is the base plate or tail part. At the end of the tail part it consists of base plate. So this base plate is attached to the collar of the collar side, and this base plate consists of 6 tail fibers. Base plate consists of 6 tail fibers and 6 tail pins. 6 tail fibers and 6 tail pins. So these tail pins functions is the tail pins have mainly 2 specific functions. One of the function is that it will attach the host of the bacterium cell. It attached the tail pins is attached the host surface of the bacterium cell. Attached the host surface of the bacterium cell. Host surface. And next one is that these tail pins are useful for the secretion of the enzyme, secretion of viral enzyme, that is the lysozyme. Viral enzyme, important. Lysozyme is the viral enzyme. So the tail pins are useful for the secretion of viral enzyme during the bacterial multiplication or during the attachment of the host cells. The tail pins have mainly specific 2 functions. One of the function is the attachment of the host surface of the body. So, and next one is the it secrete viral enzyme, that is the lysozyme. Secrete viral enzyme during the attachment of the host cell. And next 6 tail fibers. These tail fibers are useful for the attachment of the host surface of the body. So these are the structure of the T4 bacteriophages. So once again, T4 bacteriophages. Phase means virus. Virus infect the bacteria is called the bacteriophages. These bacteriophages generally infect the E. coli bacterial cells. So that's why it's named as a coliphages. Coli. Phase means virus. Coli means E. coli cells. So bacteriophages are generally infect the E. coli bacterial cells. That's why it's named as a coliphages. These coliphages is classified into 2 types. T even phases and T odd phases. Even phases, even numbers. Example, T even phases T2, T4, T6. That means even numbers, etc. And T odd phases means T1, T3, T5, T7. That is the odd numbers. So in this video, we are studying the special details of the T4 bacteriophages. This T4 bacteriophages is the structure of T4 bacteriophages. This T4 bacteriophage shape is the tadpole shape. T4 bacteriophage shape is the tadpole shape. These phases, these bacteriophages consist of 3 parts: head part and middle collar part and tail part. End of the base of the tail part: head part, collar part and tail part. First see the head part. Head part is a hexagonal head. It consists of a hexagonal head. The size of the head is 95 into 65 nm. Size of head is 95 into 65 nm. Nm means nanometers. In this 55 micrometers long double-stranded DNA tightly packed inside the head. Here head consists of a genome. That genome is the double-stranded DNA. Important. In T4 phases, genome is the genetic material is the double-stranded DNA. Inside the head, it consists of double-stranded DNA. It is tightly packed. It appears like a yarn of ball. In this genome consists of 1530 nucleotides. In this genome consists of 1530 nucleotides. This genome surrounded by protein coat is capsid. Protein coat is the capsid. These capsid protect the genome and give shape to the viruses. And this capsid is again made up of protein subunits, that is called the capsomeres. Protein subunits is called the capsomeres. In this D4, these capsomeres number is different in different phases. So that's why here T4 phases consist of 2000 capsomeres surround the capsid. And next, the middle part, it connected the one of the end of head and one of the end of tail. It is the collar. Its collar is joined by head and tail. This collar is again covered by sheath, proteinous sheath. Collar is joined by, covered by proteinous sheath. This sheath protect the collar. And here these proteins are arranged in 24 rings. Proteins are arranged in 24 rings. Each ring contain again 6 subunits. Here protein subunits arranged in 24 rings. Each ring consists of 6 subunits. Totally how many subunits? 144 subunits are arranged in the proteinous sheath. Okay, and next third part is the tail part, end of the tail part. This tail part contain one of the base plate and tail fibers and tail pins. 6 tail fibers and 6 tail pins. So here tail pins have mainly specific 2 functions. One of the function is the tail pins are useful for the attachment of the host surface cell during the multiplication, bacteriophages multiplication. Tail pins are useful for the attachment of the host surface of the body. And next, these tail pins are useful for the secretion of enzyme, viral enzyme, that is the important lysozyme, secretion of viral enzyme, lysis of the bacterial cell wall. So here 2 functions. Tail pins is helpful for the attachment of the host surface of the body. And next one is it secrete enzyme is the lysozyme. This enzyme is useful for the lysis of the bacterial cell wall during the attachment of the host cell. And next one is the tail fibers. These tail fibers are useful for the attachment of the host cell. It is helpful for the attachment of the host surface of the body. So these are the functions of the tail fibers and tail pins. Here important enzyme is the lysozyme enzyme is the viral enzyme. Lysozyme enzyme is the viral enzyme. In T even bacteriophages, 5-hydroxymethylcytosine. T even bacteriophages, 5-hydroxymethylcytosine are present in place of the cytosine. In place of the cytosine, it is only present in the T even phases only. It is absent in T odd phases. So once again important, 5-hydroxymethylcytosine, one of the base, is present in place of the cytosine in even phases only. It T odd phases, absence of 5-hydroxymethylcytosine. Here important 5-hydroxymethylcytosine is only present in the T even phases, and the viral enzyme is the lysozyme. And important question is the T4 bacteriophages shape is a tadpole shaped and their genome is the double-stranded DNA. It is a 2 marks question for the inter second year students. What is the shape of T4 bacteriophage and its genome? T4 bacteriophage shape is the tadpole shape. Its genome is the double-stranded DNA. Important for inter second year students. These T4 bacteriophage is 4 marks question for inter second year students, and it is 5 marks question for degree first semester students. Here 4 marks question for inter-secondary students and 5 marks question for degree first semester students. 5 marks question and bacteriophage multiplication 10 marks question for degree first semester students. So if you like this video, please subscribe. If you have any doubts in this topic, please comment in the comment section. Thank you so much for watching this video.